that says, on the back, that says, during the oxidation of glucose, what enzymatic steps yield CO2, and where is the O2 consumed, right? And actually, the part about the enzymatic steps that yield CO2, most people got right. And so there are three of them. Pyruvate dehydrogenase, isocitrate dehydrogenase in the citric acid cycle, and alpha ketoglutarate dehydrogenase in the citric acid cycle. So those are the three reactions that generate the carbon dioxide in the complete oxidation of glucose. Now, some people said, also threw in pyruvate decarboxylase in the fermentation pathways, but the fermentation pathway isn't really the complete oxidation of glucose like it's in there, because that product ends up being ethanol by the time you get to the end, not CO2. Where a lot more people miss, though, was oxygen, right? And what's the correct answer? You want to tell me?
ETC.
What's that?
ETC. Complex
4 of the electron transport chain. But this is a kind of a common misconception, and I think the misconception comes out from, if we take this equation that you see in biology, we started out glucose plus 6 oxygen goes to 6 CO2 plus 6 water. This thing is stoichiometrically correct, but it's mechanistically really misleading. Because you look at this and you go, okay, I've got the 6 oxygens here, I've got 12 oxygens here, and then so I have 6 oxygens here. So some of these oxygens here must be coming from that oxygen, right? If you do that. But that's not actually what happens at all. What this really is mechanistically is the result of two separate processes. The first process is the oxidation of glucose to carbon dioxide in glycolysis, pyruvate dehydrogenase, and citric acid cycle. So that process is you take C6H12O6 plus 6 waters plus 10 NADs plus 2 FADs, and you end up with 6 CO2 plus 10 NADH plus H plus. I'll do it like this, like that, and plus 2 FADH2. So if you go through and you look at all those reactions in glycolysis, pyruvate dehydrogenase, citric acid cycle, there's actually 6 waters that end up coming in, right? And so the oxygen that come into carbon dioxide are coming from the oxygen that was in the original glucose plus water. Okay, so this first phase, so this is glycolysis slash pyruvate dehydrogenase slash the citric acid cycle. That's what generates. The second phase, which is the electron transport chain, takes our 10 NAD H plus H pluses plus our 2 FADH2 plus 6 oxygens, and it ends up generating 12 waters plus 10 NADs plus 2 FADH or FADs. So this process here is electron transport. So all the oxygen that is used gets consumed in the electron transport chain. So the CO2 that's generated actually has nothing to do with the oxygen that we actually bring in. And so, if we sum all of this from a stoichiometric standpoint, we cancel all of this stuff out. And we end up canceling 6 waters out here, making this go to 6. And that's what leaves us stoichiometrically to 6 C6H12O6 plus 6 CO2 goes to, or plus 6 O2 goes to 6 CO2 plus 6 water. All right, so you see how this is a little bit misleading, right? And so the point is basically, one way of sort of saying it is this part here is glycolysis, citric acid cycle, PDH, pyruvate dehydrogenase. Here, this part here is the electron transport. Anyway, I wanted to walk through, because one of the things that, in this class, you guys are, different people are going to get different things out of the class. But one of the things I'd like for everybody to get out of this class is to answer the question of why do we breathe, right? We're doing it all the time. We know, well, we're taking in oxygen and we're bringing up, well, what is that oxygen actually doing? Well, what that oxygen is doing is going to complex 4, the electron transport chain, and basically sucking the electrons out of NADH and driving that electron transport chain so it can make a bunch of ATP. That's what the oxygen is doing. The CO2 is coming from pyruvate dehydrogenase in the citric acid cycle, and which ends up generating those high energy electrons that feed into the electron transport chain. And again, part of that is because that's what drives so much physiology, right? I've said this over and over again. That's why we have lungs. That's why we have a circulatory system. It's because of that reactions at complex 4, the electron transport chain. I mean, think about it. Think about it. What's the other role of the heart other than pumping those blood gases around? It's love, right? The heart is love, right? If we didn't have complex 4, the electron transport chain, there would be no heart. There would be no love, right? So it's pretty important. You can take your significant other and look into their eyes and say, "Oh, I'm so glad there's complex 4 in the electron transport." Start. Anyway, so that's the I just kind of really want to reiterate, and I'll give you a little hint. There's probably going to be a question pretty similar to that on the final, just because I think I'd like to have everybody walk away with the answer to that question is, why do we breathe? Why do we breathe? Okay. Any other questions on that? Who just walked in? Somebody just walked in? Last name again? What's that? Oh, do you need a calculator? No calculator for the final. Keep it simple. Actually, let me bring up. Yeah. Oh. Yeah, just kind of after class.
No, because you know it's just theoretically, right? We also want to okay, so either way I would have gone. Excellent.
All right. Well, I'll get this going. If you want copies of your other old exams, come up and see me right after class. What else? I do have a meeting from 1 until 2, but I'll be available after 2. And remember, we have the help session at 7. See what else was I going to? Oh, here's a couple of other things to help you out. If something happened and you aren't able to do a homework, and then you got locked out of it, let me know, and I'll open it back up for you so you can go in and complete that homework. The other thing is get everything. So Monday is when I'm going to basically be wrapping everything up. So we'll take the final tomorrow, and I'll grade that over the weekend. You have the double helix worksheet due on Friday, and so I'm going to be wrapping everything up on Monday. So again, if you have some of the homework or something that you have to wrap up like this, make sure to get it all done basically by Sunday night. Because then Monday I've got to actually get the grades calculated and in. All right, so what we're going to do is I'm going to talk a little bit about lipid biosynthesis, and then we'll talk about amino acid, proteins, amino acids. It's not really just amino acid catabolism. I was trying to kind of get the highlights of several different things together. Okay, there's the homeworks that are due, the final exams, okay, the pathway maps, So I would draw pathway maps for glycogen degradation and glycogen synthesis. So we did that yesterday, right? And then also for the lipid oxidation, I'd start in the cytosol and then go into the mitochondria. And then the lipid synthesis that we'll do today in there. The other thing, it's going to be over in, I got to send a notice. It's going to be over again in the in Mendocino 3013. Any other questions about this? Yeah, Lily. No, not really. I don't care. I mean, try not to make it a book. Yeah, yeah.
Since it's a final, will you get the whole period to take?
Yeah. You'll get a couple hours. You can relax and take your time. I mean, I won't write it like a 2-hour exam, but it'll be 120 points, so it'll be a little bit longer than, say, the last one that you took. You'll have the whole time. Yeah.
Is it 125 points or 120 points?
I thought I had it set at 120 points.
So in the syllabus it's 120, and then in our Canvas it's 125.
I'll double check, but it'll be one or the other. I'm pretty sure 120 is what I thought in the syllabus. I had to adjust it to get it like a nice even number of 600 or something like that. Anyway, that'll be something. Let's see if there's anything else. Yeah, just make sure again, especially for those of you who are kind of on the margins, make sure you're getting the homework done and getting the getting the homework done and doing the double helix sheet. Typically those scores are pretty high, so I'm kind of counting on that to help get people go over a little bit of a hump. I do still have to check off that you did the pathway maps and get that going. And again, if you're on the margins, try to spend a little bit of time today studying, and so that the final exam helps you rather than hinders you. Any other questions on that? All right, so what I'm going to do is I'm going to start with a few slides, and then we'll get into a little bit of the board work with some pathways. So we're talking about lipid biosynthesis. So lipid biosynthesis in vertebrates occurs in the liver primarily, and one of the ways you can illustrate that is if you take fowl like ducks and geese and you force feed them grain with high carbohydrate meal, okay, they'll have excess carbohydrates, and the liver will convert those excess carbohydrates to fat. And in fact, what the liver does is it swells up as it accumulates all the fat from being force-fed all those carbohydrates, And then what people do is they harvest those livers and they make the delicacy foie gras out of it. So that just is a way of illustrating that fatty acid biosynthesis, triacylglycerol biosynthesis happens in the liver. Now, waterfowl aren't the only things that are funnel fed sometimes. Beer in humans like this, they can get that. So in here we have metabolism of alcohol, right? And if you have the metabolism of alcohol here is ethanol to ethanal, you make some NADH, ethanoate, then you make acetyl-CoA. You basically make acetyl-CoA. Now if you have plenty of energy needs that you can convert that acetyl-CoA enter in the citric acid cycle, and it can make some ATP and like this. But if you don't have that energy demand, then you're going to store it away in the triglyceride biosynthesis. And again, that tends to happen in the liver. And so what happens basically is you generate those lipids and you get the infamous beer belly. Problem is that I've been using that slide for about 10 years. I'm getting more and more to look like that guy. Anyway, but the other thing that can happen from too much alcohol consumption is the condition known as fatty alcoholic liver. And so that's a stage on the way to cirrhosis, basically when you get large fat deposits that are actually building up in the liver. And again, this is the site of triacylglycerol biosynthesis. And if you look at a micrograph, you can see the tags, the triacylglycerols basically accumulating in the liver. All right, so let's go think about, we saw the structure of fatty acids and the triacylglycerols, and for this today, we're just going to focus on the biosynthesis of fatty acids, and then those fatty acids, there's a whole other series of reactions that basically link it to the glycerol backbone to make the triacylglycerol. But here we're just going to focus on the biosynthesis of the fatty acids. And we saw when we degraded fatty acids, what we did is we chopped 2 carbons off at a time and we turned those into acetyl-CoA. Well, the synthesis of those is basically the reverse of that. You're going to take acetyl-CoA molecules, link them together to build up a fatty acid. And so this is a process that takes place in the cytosol. So remember all our acetyl-CoA, what's the location of pyruvate dehydrogenase? What's the subcellular localization of pyruvate dehydrogenase? That'd be a good question for the final, I guess. Like this. Pyruvate dehydrogenase, what do you think? The matrix, the mitochondrial matrix. Pyruvate dehydrogenase, citric acid cycle, all in the mitochondrial matrix, right? So, but fatty acid biosynthesis is occurring out in the cytosol. So we had to figure out some way to get the acetyl-CoA that's generated by pyruvate dehydrogenase in the mitochondrial matrix out to the cycle. And it's kind of a convoluted process. It's basically, you use citrate as a transport molecule. So here's our mitochondrion, and here's our oxaloacetate, and then the first enzyme of the citric acid cycle, citrate synthase, is basically going to make citrate, where we take the acetyl-CoA, link it to oxaloacetate to make citrate. The citrate could either keep going through the citric acid cycle, or if there's not any demand for that, it can be exported through a specialized transporter here out into the cytosol, and then it undergoes a reaction known as ATP citrate lyase, in which you end up cleaving off the 2 carbons to remake acetyl-CoA and oxaloacetate. Here. And so now we have a cytosolic source of acetyl-CoA. Okay, so then the next part, let's see, I think this is where I'm going to go to the board here, but the next part is basically taking these 2 carbons and polymerizing them, adding them 2 molecules at a time to end up making the fatty acid. Yeah, so the overall reaction of that is to take acetyl-CoA, and that what I'm going to talk about in just a sec is what you end up doing is activating the acetyl-CoA to make malonyl-CoA, polymerizing, and then you elongate it to palmitic acid. So we're going to go over these reactions, and part of the reasons for wanting to go over that is because you'll see they're related to the same reactions of beta oxidation and those that we saw yesterday, as well as those reactions of the citric acid cycle that we talked about yesterday. Okay, so this is fatty acid biosynthesis. And let's see, I'll kind of summarize some of those things in the overview. First, we'll say it occurs mostly in liver. Secondly, occurs in the cytosol. And I guess a subpoint of that is that acetyl-CoA is transported out of the cytosol, out of the mitochondria via citrate and then the action of ATP citrate lyase once it gets out in the cytosol. All right, so now we have our acetyl-CoA out in the cytosol. So the next part we'll say is the activation of acetyl-CoA. So again, kind of like we had with when we were making glycogen yesterday with glucose units, and then we couldn't just easily link them together. We had to activate it. We had to make the UTP glucose pyrophosphorylase in order to be able to, or the UTP glucose in order to be able to have enough energy to form the glycosidic bond. Well, the acetyl-CoA also has to be activated. And the way it's activated is by carboxylating it. So we take acetyl-CoA and we bring in carbon dioxide and we end up making this compound here, which is known as malonyl-CoA. And this requires the hydrolysis coupled to the hydrolysis of ATP. And the name of this enzyme is acetyl-CoA carboxylase. And it has an interesting mechanism. In other classes, we delve into the mechanism of that. But given that this is what the day before the final, we will not get into that mechanistic detail. But I do want to make point is that this is a major regulatory point for fatty acid biosynthesis. Also has a biotin cofactor. All right, so now we have this activated form of acetyl-CoA, the malonyl-CoA. We can begin to polymerize those acetyl-CoA units. And this is done in a big multi-enzyme complex known as fatty acid synthase. So the first thing that we have is So we have our acetyl-CoA, and then we have a molecule of malonyl-CoA. The first thing that happens is these molecules gets transferred to this big multifunctional enzyme. So these are coenzyme A thioesters. So basically on the enzyme is some pantothenic acid and the thiols that are very similar to CoA, but that are covalently attached to the enzyme. So basically the enzyme has the business end of CoA already attached to it. The part of that enzyme that, of this big multifunctional enzyme that has that is known as acyl carrier protein. So what we do is we come in, we bring in this acyl carrier protein, and that's abbreviated ACP, and we drop off coenzyme A, and now we have, all we're basically doing here is we've transferred this acetyl group now to the enzyme. I'm going to write SACP, because now it's actually part of this multifunctional enzyme. And the same thing happens over here. So this was acetyl-CoA here. This is acetyl ACP, acetyl acyl carrier protein. This is malonyl-CoA. And this is malonyl ACP. So not too much interesting chemically going on there. We're just basically attaching it to this big actual multifunctional enzyme. Now the next step starts the polymerization reaction. I'm going to put a little star by that CO2, because basically now what we're going to do is we are going to so these two carbons here basically get added on to the end here, and we lose this carbon dioxide. So this is known as the condensing enzyme. This is the beginning of the polymerization process. And then this compound here is known as beta keto acyl ACP. What's that?
Does the sulfur leave too, or the malonyl leave?
Yes, actually that's good, thanks. So then this is SHACP comes off, yeah.
And I guess we don't need any of the enzymes.
Okay, so the enzyme names is these are trans-acylase enzymes. This one is acetyl-CoA acyl carrier protein trans-acetylase. That one's malonyl-CoA acyl ACP. So I'm just going to put trans-acylase here. This one though, this one is a good one. So this is beta keto acyl ACP synthase. It's also known as the condensing enzyme, or Cas. right, now what I want to do is, now we have this beta-ketoacyl ACP synthase. But a fatty acid doesn't have a bunch of oxygens in there. We basically have to get rid of that oxygen. We have to reduce that oxygen. So we have a beta-keto group here. This is just like, again, oxaloacetate had this beta-keto group. If we basically run essentially the citric acid cycle reactions backwards from oxaloacetate back to succinate, we'll see that that basically gets rid of that keto group. And that's essentially what this next set of reactions are going to do. So we're going to, we've got our here's our beta-keto-acyl ACP synthase. And so here, I'll draw over here sort of the analogous this was our oxaloacetic acid intermediate in our citric acid cycle, okay? And we got this oxaloacetate by oxidizing malate. And when we did this, we took NAD and we made it NADH. This is like the last reaction of the citric acid cycle. Okay, now we're going to basically do this in reverse. We're going to come in and we are going to take, we're going to reduce this, so we're going to need some high energy electrons. We're not going to use NADH. This is an anabolic process. We're going to use NADPH. Here's a quiz for you. What pathway in heterotrophic organisms like us, what pathway generated that NADPH that I mentioned briefly yesterday? The same pathway that also generates the 5-carbon sugars for like nucleic acids. Yeah, 5, remember 5 pentose, the pentose phosphate pathway. Maybe I'll do that as a little reminder from pentose phosphate pathway. Okay, so now what we're going to do is we're going to make the alcohol here. This enzyme is beta-keto-acyl ACP reductase. And now this is beta hydroxy acyl ACP. Again, so back here in our citric acid cycle, which goes in this direction, okay, this is the citric acid cycle, we got this compound by taking fumarate and adding water to it. That's how we got this. So we're going to basically do the same thing backwards here. We're going to remove water here and create a double bond. this molecule here is and let's see, this enzyme that does this is the beta hydroxy acyl ACP dehydrase. Because we're removing the water to create the create that double bond. All right, so we still have this double bond, but a fatty acid is all saturated chains, right? Well, again, reverse of the reaction to the citric acid cycle. The way we got our fumarate here was by taking succinate and doing the succinate dehydrogenase reaction, FAD to FADH2. So we're basically doing the same thing here. Oh, I forgot to put my hydrogens here. We're going to do the same thing here, except now we're going to use for our high energy electrons NADPH. And we're going to generate now the 4-carbon saturated acyl carrier protein. So we've gone through these reactions. Oh, and then this reaction is enoyl ACP reductase. So now we have a 4-carbon chain, basically a 4-carbon fatty acid. We've condensed, overall we've tensed 2 acetyl-CoAs and made basically the 4-carbon version that's totally saturated here. Now, but you know, our fatty acid's going to take 16 carbons, right? So we have to keep this process going. So in order to keep this process going, this actually will now sit at that site here. So instead of at that site having acetyl ACP, it now has this butyryl ACP. And the whole set of reactions will continue on, except this will have 4 carbons. We're going to add, this will have 6 carbons with the keto group. We'll go through all this reaction and we'll end up with a 6-carbon version. And then we'll come back and we'll do it again, we'll do it again, we'll do it again, we'll do it again. Up until we get 16 carbons.
What is this, the name of this one?
What's that?
What's the name of the one at the bottom?
This one is butyryl butyryl ACP. That's what that molecule is.
Okay, thanks.
And now it basically becomes a substrate in this, for this reaction here, the condensing reaction, to now take it from 4 carbons to 6 carbons. Again, it kind of, the whole cycle keeps recycling. Well, how does it stop, right? We just keep adding until we have this great big huge one. Well, it stops at 16 carbons. And so the final enzyme of this fatty acid synthase So now we end up having after multiple cycles here, we end up having 2, 4, 6, 8, 10, 12, 14, 16, like this. This is ACP. So again, after, let's see how many cycles. This is going to be 1 cycle, 2 cycle, 3 cycle, 4 cycle, 5 cycles, 6. After 7 cycles, we get palmitoyl ACP. And there's an enzyme known as a thioesterase, which just takes water, cleaves off the coenzyme A, and makes palmitic acid. And that's the end of the fatty acid synthesis. And this is this enzyme is called thioesterase.
Can I ask about this step here with the enol ACP reductase? Usually it goes to NAD+, right? But what's going on there?
Oh, yeah, thanks.
Thanks.
Now, this is as far as much detail I think we'll try to get into this morning. Now, of course, you guys know that all fatty acids aren't palmitate, right? A lot of them have 18 carbons, a lot of them have double bonds in them and all that sort of stuff. So what happens there? Well, it turns out that there's that the main fatty acid synthase generates palmitate, and then the palmitate will enter the endoplasmic reticulum where there's another elongation system which will add another 2 carbons to make it 18 carbons long, and also in the endoplasmic reticulum there's some desaturases which will actually introduce double bonds in the molecule. We won't get into there. And then once you have all these fatty acids of the appropriate length, and there's a whole other pathway that basically takes the fatty acids, transfers it to the glycerol backbone to either make your phospholipids or your triglycerol. That's sort of the rest of the pathway. But I wanted to at least go through this cycle just to reiterate is that series of reactions that we saw in the citric acid cycle, we've seen that pattern again. So like in the citric acid cycle forward direction for beta oxidation of fatty acids, and then for fatty acid biosynthesis, it's basically those reactions reversed in there. I mean, it's not the exact same enzymes, but it's the same biochemical approach. The other thing I want to mention about this pathway is this fatty acid synthase enzyme, because it is pretty cool in terms of being a big multifunctional enzyme. So it's actually a dimer, and this is one monomer, and you see it's a great big polyfunctional enzyme. When all these enzymes that we've talked about, here's acyl carrier protein right here. Here's the thioesterase at the end. Let's see, this is the condensing enzyme. This is the acyl transferase that we had. This is the dehydrogenase, the enol reductase, etc. All those enzymes are basically all connected kind of in a chain or whatever on the same single polypeptide. So this is actually known as a type 1 fatty acid synthase. Now, if there's a type 1, there must be a type 2. What's a type 2? The type 2 occur in bacteria. They also occur in plants, in the chloroplast of plants. That's a whole other story. In plants, the fatty acid biosynthesis actually occurs in the chloroplast rather than in the cytosol. But in bacteria, all these enzymes are separate enzymes, just like they are in, like our glycolysis enzymes are separate enzymes. But in most eukaryotic systems, those enzymes have actually fused into a single a single polypeptide with different catalytic domains on the same polypeptide. And you know, some of the evolutionary advantages to that is then it's easy to co-regulate. You know, if you want to have, you know, a stoichiometric amounts of these different enzymes to be able to drive this whole process, plus do some substrate channeling, and it's like channeling. If you fuse this into a single polypeptide, then you're always going to have the same amount of each of the required enzymes, and the substrate's not going to get very far. The substrate attached to this acyl carrier protein is going to be right accessible to all those different different activities. So you think, okay, well, that kind of makes sense. But on the other hand, bacteria and plants are pretty successful, right? Who's to say that this is a more advanced evolutionary thing? So, you know, that always becomes a little bit of philosophical questions. It's like, okay, if this is such a great advantage, how come bacteria didn't fuse their genes to be able to make a multifunctional fatty acid synthase? Here's a couple of other things. So we talked about how all these reactions occur attached to this acyl carrier protein. This is the structure of coenzyme A I showed you before. It's basically ADP plus this pantothenic acid thing, and then at the very end is this thiol, like this. So basically, this phosphopantothene is basically now connected, that ultimately is attached to a serine residue on the protein. But it basically then can attach those acetyl-CoA and those other CoA thioesters. Yeah, that's just here, this is the genes that this is the arrangement of the different domains on the polypeptide chain. So here are different, here's the amino terminus, and then the carboxy terminus overall, and we have the condensing enzyme, the acyl transferase, dehydrogenase, or basically all those different domains are all linked together. And presumably they arose by gene fusion, that basically all the stop codons in between this individual genes were mutated away, so then it ended up becoming a single polypeptide. Here's just another kind of view of the arrangement of that multifunctional polypeptides. Okay, let's stop there. We'll take a break. And actually, I'm going to take a break, but I'm going to ask you guys to do something. If you haven't done it already, on your devices, go to the Canvas page and fill out the course evaluation, okay? So go ahead and take 5 or 10 minutes. I'll give you a little bit of extra time to get on your device if you haven't done on there.